at the Mayo Clinic. And now we have the opportunity to listen to his work here. So this is going to be a very exciting seminar. Alex Gaspar Maia, he's an associate professor of the lab of medicine and pathology at the Mayo Clinic and the director of the epigenomics development lab for the Center for Individual Medicine at the Mayo Clinic. He earned his PhD in molecular biology from the University of Coimbra in Portugal, while performing his thesis project at UCSF in STEM cell biology in the lab of Miguel Ramlo Santos. For his postdoctoral training, he moved to Mount Sinai, New York, to the lab of Emily Bernstein to study the role of histone variants in several aspects of reprogramming and cancer. In 2017, he started his own research group at the Mayo Clinic. The Laboratory of Functional Epigenomics has been working on various aspects of transcription and enhancer regulation with implications in cellular heterogeneity and drug resistance in cancer and cellular reprogramming. The goal of his lab is to use epigenomic profiling to better understand cancer programs associated with malignancy, metastasis, drug sensitivity, and define transcriptional dependencies. He has received numerous awards, namely 2 postdoctoral fellowships from the USA Department of Defense, New York Stem Cell Foundation, the NIH Career Enhancement Award for the Mayo Clinic Ovarian Cancer SPORE, and from the Breast Cancer SPORE, and also the Young Investigator DoD Ovarian Cancer Academy Award from 2021 to 2025. So it's really my pleasure that he's here, and the floor is yours.
Thank you. Is this good? 1, 2. All right. Thank you so much, Maia. It's really lovely to be in Mexico City. I guess I got a little too excited about Mexican food and I started eating a lot of street food. And today I'm not feeling super sharp, but I hope it's all your guys' fault. The food is lovely though. So today I'm going to be talking about a recent story that we actually just submitted. But before I get into that story, I actually kind of wanted to give a little bit of an overview of my path and my career. I know that there's some graduate students here and, fellows and postdocs. And so it's always nice to have a perspective on things. Also because, my trajectory was not very, what would one say, standard. I actually started as a marine biologist. That was my undergrad. In Portugal, the undergrad was 5 years. So it was kind of like a mix of 3-year plus 2 of masters. And at the end of my undergrad, I basically decided to do my thesis on molecular biology. And it was kind of interesting because before, I thought I was going to save the oceans and the whales and the dolphins and all that. But then when I actually got into the lab, and probably also because I had an amazing mentor, I was really fascinated about changing things molecularly and doing that very, very easily, which is something that you can't do with the oceans, right? That you can't move whales or dolphins from one ocean to another and see what happens. You can't put cold water here and hot water there. So the ability of doing this molecularly really fascinated me. So I basically kind of shift gears and decided to study molecular biology. During that time, I was also working as an environmental educator. So, my fields were kind of still open in some ways. But then I was actually really lucky to get into this PhD program from Coimbra, as mentioned. That actually was an amazing program because it was give us a fellowship that we could use to go anywhere in the world. So all I needed to do was send an email to PIs in different labs and ask if they had a spot. And luckily, Miguel Ramalho Santos, who was also Portuguese, but had just started his lab at UCSF, took me under his wing, and I did my PhD all in San Francisco. And at the time, I was really fascinated about cell identity, cell fate. And initially, we wanted to really just look at genes that were potentially important for the cells to stay pluripotent, to have the ability to differentiate in all kinds of cell types. And this was kind of like what led me to really become really fascinated about the nucleus, chromatin organization, and epigenetics as a field, because what we actually end up finding is that the majority or a very important part of the identity of the cells really comes from the epigenome. And because of that, then I decided to do a postdoc at Mount Sinai. with Dr. Emily Bernstein, and I really, wanted to work alongside some of the leaders in epigenetics. Emily Bernstein had just been a postdoc with David Alice, which is one of the fathers of epigenetics, or was, because he passed away last year. And then we studied a variety of different topics, but also another important thing about my path is that I actually, when I started, did not want to work in cancer. And that was really not my perspective, especially, after being a marine biologist and after being, after starting to work on cell identity and cell fate in early embryonic stem cells, I was really fascinated about these initial and basic questions about cell identity. But my PI actually, when I had just started, said, why don't you apply to this grant? There's this funding mechanism to work on breast cancer. Our lab next door was working on breast cancer, so he became my co-mentor, and I got the grant. So then I actually had to work on breast cancer for 3 years or so. And then I actually got to understand that actually, even though cancer becomes really challenging, especially looking on chromatin because the cells are all messed up, as you probably know, it is also really interesting in terms of understanding cell fate. And what's even more fascinating is that a lot of the pathways that are used for in cancer are basically hijacked from things that we know in development. So that link was very fascinating to me. And then I started my lab in 2017, and really with this idea of try to understand how we can learn from chromatin and apply that to cancer. Currently, the lab has a lot of the funding comes from breast cancer, in ovarian cancer. But we actually, during the pandemic, the lab was able to actually stay open for the most part because we started doing single cell analysis in patient samples. So as you can see, if you're feeling maybe my path seems a little crazy, everybody's path is potentially crazy if you actually get to talk to them a little bit. And so good luck with all of that. So, I also bring this up, I'm sure a lot of people here talk a lot about epigenetics. Some of you folks probably not so much. So I usually use this in my class in genome biology just to kind of prime everybody to at least the way I think about epigenetics or chromatin biology in general. So this, as you probably can guess, is a part of our genome, a very, very small part of our genome. And, for a cell to actually do its job, it needs to take information from this big group of nucleotides and start choosing specific regions to start, transcription, right? So one thing that the epigenome is very important at doing is annotating this big book, which is the DNA that we carry in our cells, and annotating at the regions that are more important for protein coding to code for proteins. Also very importantly, it needs to know which way it goes because, as you probably know, it's a double helix. So some genes actually get expressed on the 5' end or reverse, and so the epigenome or chromatin marks actually are very important to define where the promoter is, or in this case, TSS for transcription start site. I forgot to mention SOX2. It's one gene that I annotated here, which actually is part of one of the stories that we're interested in. Also, very important is that, as you probably are aware, most cells, with a very few exceptions, carry exactly the same genome in our bodies. But so in order to actually have different identities, muscle cells, neurons, blood cells, and all those, you actually need to select the genes that you are expressing. And so the epigenetic marks really allow us to define whether a gene is going to be activated or repressed. Obviously, there's a lot of pathways and there's a lot of mechanisms to activate genes, but then what's very important in the way that chromatin is organized is that we keep some of the genes silent. And that's, part of the epigenetic code. And then also importantly, and this is something that we are really interested in our lab, is to look at these regulatory elements. So regulatory elements are typically regions where transcription factors bind to. In this case, this is, for example, a SOX2 binding site of its own gene. And so these sequences, these transcription factors are recognized as very specific sequences, and they basically, that binding leads to either activation or repression. And that's also important because not all enhancers really determine activation, they can also determine repression. And by us understanding all these different connections, it will, potentially will allow us to understand how some genes, some cells are specifically differentiated into muscle cells, or in the case of cancer, do they actually, some of these cells are really addicted to some of these enhancers or not? And so this concept is what we're trying to follow in my lab. This again, it's kind of like just an overview of, people always show a slide like this to kind of like highlight the fact that, you know, the genome that I was just presenting to you, which should be these molecules here, is much more complex than we think when we illustrated like I did. And so, for example, there are modifications of the actual nucleotides that, you know, lead to changes in expression, such as methylation. And then the histones, which basically are these proteins that have the DNA wrapped around them, have these tails that can be modified, and with some of the residues really have a very specific meaning. And those are the colors, the color code that I was showing you before: red for repressive, green for activation. Those are just illustrations. Obviously, that's not how genes are repressed in the cells. But so if you, for example, pull together some of these marks, what we call histone marks, they usually have very specific patterns. And these patterns really allow us to, even just looking, if we look at just the patterns themselves and forget about the nucleotides, you can actually tell whether there's a gene being expressed there, whether there's an enhancer activated, whether there's a promoter. So you can really just look at the color of the genome and understand a little bit about what's happening. So a couple of years back, I actually was still following up on a story that I had started as a postdoc, and was really interested in histone variants. I don't really need to introduce you too much into detail what all those are, but we were very interested in different patterns in regulatory elements. So we mapped them using chromatin immunoprecipitation. We mapped a bunch of different marks, and we basically try to identify the marks that are specific to, for example, an active enhancer. People had already shown this before, so this is not new, but people had shown H3K27 acetylation as a mark for active enhancers. H3K4 trimethylation is the mark that we typically associate with promoters. K27 trimethylation regulated by the polycomb is the repressive mark. ATAC only, so we were looking at open chromatin regions, and some of them didn't really have any more marks, so we just called them ATAC only. And then there's this class that we identified that was basically enriched with this new flag, as we call it, called macroH2A, which is an histone variant, and that actually is associated more closely with inactive repression in terms of the enhancer activity. But so as you can see from this proportion plot here, there's close to 8% of our genome is actually regulatory elements, which brings us to a very important part of our genome. And also, even if you just think about it, this is in terms of bases. We're looking at 8% of bases. And if you see that, because what I was illustrating, the enhancers, they're actually much smaller than genes for the most part. And so if we're talking about 8% is actually the amount of enhancers or regulatory elements is actually really immense. This is a little bit different in the mouse, but also probably a lot of people mentioned this, that one of the reasons probably our genome is so complex is that actually not so much in the genome itself, the letters, but actually in the complexity that comes with it by these regulatory elements. As I was alluding to, there's a lot of different layers that we can use to understand enhancers. Histone modifications, as I was talking about, K4 trimethylation, K27 trimethylation, and so forth. Histone variants like macroH2A is an histone variant. All of those add little flags, and their patterns are very specific for us to understand what they mean in terms of expression. DNA methylation patterns, and then one important part of all this is, like, once you identify the regions that you are interested in, a subset of regulatory elements, what are the transcription factors that actually bind to them? And this can be done, in silico by approximation of, like, what are the most likely transcription factor motifs that are incorporated in those regions, or we can also do, look at the binding specifically by chromatin immunoprecipitation. And then all of these, or most of these enhancers, actually, if they're active, they also express non-coding RNA. So that's also another pattern that people can follow up on. So when I was a graduate student, as I mentioned, I was working on stem cells, and actually my project was really try to understand cell identity and why an embryonic stem cell really is able to differentiate into all kinds of cells in the body. And we initially started to look at different genes that were important. So we were knocking out different genes and try to see if the cells would complain and stop being pluripotent. And then right around the time that I was starting this project, a group in Japan, Dr. Yamanaka, published this very important paper showing that just adding 4 different transcription factors, you can actually turn any different cell. Here, the paper was just on skin fibroblasts, but pretty much at this point, every single cell, different cell type, can be transformed into an embryonic stem cell. And in the mouse, you can actually do that and generate a whole mouse just out of this approach. So knowing that only 4 transcription factors could do that really changed the way we understand biology. So my contribution for that was really, as I was mentioning, was about the chromatin, and I was really interested in how the chromatin was organized. And so we actually identified chromatin remodel CHT1 that was really important and involved in changing the way the chromatin is organized. And so that was kind of like how I started really becoming fascinated about chromatin and epigenetics. And then soon later, I started becoming interested in how the epigenome is affected in cancer. And this is a very nice illustration that I just saw a couple of weeks ago from Nature Genetics, showing basically this is the epigenetic landscape as people typically show it. And you have an embryonic stem cell or a pluripotent stem cell up in the top, and then it has to make decisions and goes through these values for differentiation purposes. And what they actually are trying to illustrate here is that some of these things break in cancer, and what happens is that instead of differentiation, you get activation of programs that are really malignant. What we are trying to understand is can we understand this path, and can we actually block it at some point, either early on, and that would be ideal, but also in terms of therapeutical approaches. And that's really the concept of transcriptional dependencies that we like to study in the lab. To look at the epigenome globally, and because I started my lab at Mayo Clinic, we were really interested in looking at patient samples. We initially started looking into some techniques that were just becoming available that allow us to do epigenomic profiling at relatively cheaply, and to look at these things across different samples. And so this is a paper that came out around the time that I was starting my lab, showing that if you can take different tumor types from TCGA data set and perform this so-called assay for transposase accessible chromatin that basically makes use of this transposable element TN5, which has been modified molecularly for our purposes, that adds little barcodes to it. And so what happens is that you actually add TN5 to an intact nucleus, and if it's really well preserved, the nucleus, what this TN5 is going to have access to is regions that are devoid of chromatin or nucleosomes. And so these regions that TN5 is actually going to be incorporating are so-called open chromatin regions that really represent enhancers and promoters. And this is only possible because we treat these cells for 30 minutes, for example, and then stop the reaction. Otherwise, you would start having TN5 incorporating in all regions of the genome. And because we give it such a short period of time, we actually are able to make these little dents, these cleavages around the genome. And this basically then, because we already added some barcodes, it actually can be sequenced, and it's relatively easy. The ability of doing this in a variety of different patients really started highlighting the fact that different subtypes or different types of tumors have very specific profiles. So this is a peak here represented here is basically, it's basically an open chromatin region. And you can see that, here are represented different open chromatin regions. And those are because each tumor will have very specific requirements or transcriptional dependencies. And then you can, do a lot of different analysis. When we started this project, we were basically focusing on ovarian cancer, which had not been used in this study. But as we were, starting to look at patient heterogeneity and try to really find specific patterns that, for example, would allow us to understand how patients respond to specific treatments, we really started being interested in tumor heterogeneity within interpatient, so within the same patient. And so my lab really started shifting the interest in looking at chromatin towards a very specific single cell approach. And so what I'm showing here is basically representation of a tumor that represents a lot of different tumor cells, and also these call it in green drug-resistant cells. And these are the cells that potentially will not be able to be treated by chemotherapy. So in ovarian cancer, which is what we're going to focus on, unfortunately, the treatment has been really pretty much the same for the past 30 years with a few exceptions. And so in a way for us is a good model because all the patients actually undergo chemotherapy as a first line of treatment. And so we kind of have a very standardized way to look at patient samples. And our idea was that even though chemotherapy is actually very good at killing 95% of the cells, sometimes there's a few cells that don't die. How can we tell them apart when we are given a tumor? Like, can we actually define which cells are going to be drug resistant or not? That's the approach by using single cell approaches. That's what we were trying to get at. Yes, one question.
Just out of curiosity, those resistant cells, are they, what's the status right now in cancer? They are assumed to be adapted or selected?
Right, so we will get to that because that's exactly what we're going to try to understand. I mean, overall, globally at this point, people have tried to do whole genome sequencing and try to look at specific mutations. So, in some cases, maybe 10% of the cases of resistance can actually be explained by a specific mutation that we would not see it because we were just sequencing globally. And some mutations really allow for, the acquisition of resistance. But in other cases, or the majority, at least at this point, we actually can't tell by mutations or anything. So there's got to be other mechanisms. And that's where, the epigenetic part of the story comes into play. And so our idea was really to try to understand if we could find some kind of regulatory elements that would be specific to the cells that are going to be treated, and whether that's going to be something that already exists in the tumor before the chemotherapy or that gets acquired. It was also something that we wanted to look at. So to do that, we started in the lab using this platform called the Multiome from 10x Genomics that basically allows us to do isolate nuclei and look at expression and open chromatin from the same cells. So we are able to, from a single cell, in this case, a single nucleus, we're able to define the expression and the open chromatin profile. But this is really just kind of like highlighting that right now, the space of multiomics and single cell multiomics is really exploding. If you take a long nap, by the time you wake up, there's going to be already a new technique for you to look at. And that's really nerve-wracking, especially for students, because they're trying to understand data. So this is basically how it works. We get from one nucleus little dots that are represented here. Those are the open chromatin regions that we collect. Then if we pull them together by similarity, you can kind of get a pseudo bulk signal of ATAC-seq, which are those peaks that are typical for open chromatin regions. And then the same thing for RNA expression. And what basically we can get from this is that we have from the same cell genes that are active and open chromatin regions that are highlighted. This is just kind of like an overview of how the system works. You actually have to isolate the nuclei first, do the transposition, and then add them to the barcoded beads. Once they are barcoded, every nucleus is barcoded, then you can actually generate two different libraries, one for the expression and the other one for the ATAC-seq. And so, I mean, today I'm really going to only focus on the first part of this analysis. We call the persistent cells, and we will get to that in a minute. But obviously, because we're looking at single cell analysis, we can start inferring or trying to delve into questions about tumor microenvironment. Very importantly, ovarian cancer has not been very successful with immunotherapy, has not been very successful application in ovarian cancer. So there's definitely a need to try to understand why some of these tumors are immunosuppressive. And then another student in my lab is actually trying to understand the dark side of the genome, the transposable element, which get reactivated in cancer, but in normal cells, they actually are very nicely epigenetically repressed by DNA methylation and other patterns. All right, so just a little bit about ovarian cancer. Ovarian cancer, I mean, it's not one of the top most malignancies in women in the US, but then when you actually look at the estimated deaths, it actually ranks much higher. And the reason for that is kind of try to explain here on the right side is that the mortality for these patients is really high. So when you get diagnosed with ovarian cancer, your chances of surviving are much less than breast or uterine cancer. One of the reasons for that is it gets diagnosed much later. It's very hard to diagnose ovarian cancer. A lot of the symptoms are actually associated with a lot of things that women experience postmenopausally. So it's very, very, very common that a woman goes to the clinic and already has a high-grade tumor, which then becomes really hard to treat. And one important part of the story is that actually ovarian cancer, or in this case, I'm going to really focus on a subtype, which is the most common one, called high-grade serous carcinoma. One important feature of this is actually ovarian cancer does not, for most of the times, does not come deriving from ovarian cells. It actually comes from the fallopian tube. It actually, arguably, some people actually discuss ovarian cancer as almost the first metastatic tumor, because the cells of origin, the consensus currently is that the cells of origin, for the most part, are from the fallopian tube, and because of their tropism, they eventually attach to the ovaries, and that's when it gets diagnosed. But so in order to understand the disease, we actually have to understand a little bit about fallopian tube cells. There's two different types of epithelial cells that give rise, that are in the fallopian tube. The ciliated, represented here with those little hairy things, and then the secretory, that potentially are the big source of the origin for most of these tumors. Also, an important feature that's shown here in the bottom is that for the high-grade serous carcinoma, almost all the tumors, close to 95% of the tumors, have a p50 mutation, so it's very p50 driven. Again, very good in terms of the model system, because the homogeneity in terms of molecularly is very good for us to kind of like start looking into patient heterogeneity. One important feature is that actually molecularly there's almost what we call two subtypes of ovarian cancer, high-grade serous carcinoma. HR deficient, and this stands for homologous recombination deficient. These are the patients that have BRCA1, BRCA2, and other genes that are associated with homologous recombination. BRCA1 is a known gene. BRCA1 and 2 are known genes in breast cancer as well. And so almost half of the patients we call homologous recombinant deficient, because they have this inability to perform homologous recombination. And this has actually been explored very, in the last 10 years or so with PARP inhibitors. And so that's one of the few developments in terms of therapy, is the use of PARP inhibitors in ovarian cancer. So when we started to do this, try to understand a little bit of ovarian cancer at the single cell level, we teamed up with two major groups. Raj Kannan actually collects fallopian tube epithelium from prophylactic surgeries, or surgeries associated with non-malignant conditions. And so we have our normal control with his student Meg Ryan, and then John Waroha is the clinician that basically provided us with the samples from high-grade serous carcinoma. And then on the left side are the people in my lab that actually did the work. One important feature that we started when we started collecting these tumors is that most of them, and really this depends a little bit on the clinical side, most of the tumors currently are getting debulking surgery prior to chemotherapy. So we got 6 tumors that never seen chemotherapy. We call them naive tumors. And then 3 patients actually had already undergone a first cycle of neoadjuvant chemotherapy. And so we call these NACT, neoadjuvant chemotherapy. And these are what we call the representative of residual disease, right? Because these tumors already seen chemotherapy, and what we are going to see of them or learn from them is the fact that they already been treated. One important feature that I need to highlight is that we actually don't have a longitudinal sample pool. So all the chemotherapy naive are specific, and the neoadjuvant we don't have prior to chemotherapy. So that's one of the big problems of our data set. So, there's a lot, the single cell data can be quite overwhelming. I'm not going to really get into so many specifics. Here, this is again just highlighting the cohort. I'm going to change this again. Oh, now they're all dead. Let's see this one. Oh, that's it. It's okay. I'm just highlighting here the cohort. And if you use, as I mentioned, if we use gene expression or the ATAC-seq that's on the left, on the right side there, JAX is for expression, ATAC-seq is for open chromatin. You actually, using just those data sets alone, you can see that the patterns are relatively similar. One thing that you can see right away is that there's a lot of pink clusters. Those are the epithelial cells, and those cluster independently because they're patient-specific driven. So every patient is very different, and that's why the cells tend to cluster on their own. But then if you look at the other colors, actually most of the other colors tend to cluster together. And this is, all the T cells, macrophages, and endothelial cells. All of those are much more similar regardless of coming from patient A, B, or C. So that's really highlighting the fact that, tumor is a really messed up cell type. And then what we can actually do, because we have these two platforms, is then put them together. This is a very bioinformatic analysis, but you put them together, and now you actually have a clustering that includes both the knowledge of open chromatin and the knowledge of expression. And on the bottom, I just, the same plot is shown by patient type, so fallopian tube, naive, neoadjuvant, and then for all the patients that we collected and the number of cells for each patient. So when we, yes.
Orange and the green are about 3,000 per sample. If you multiply 5 times 3, it's about 15,000. But in the middle panel, and 6 gives you more cells.
Yeah.
Right down. So what are the controls, or what is the criteria that you use for analyzing?
Right. So we basically, because this is coming out of the surgery, we have very little control on, how much the tumor will collect, the quality. So we remove all the dead cells. And so for each group, we will have very different number of cells. The analysis, and I don't really talk a lot about the analysis, but the analysis allows us to actually randomize numbers of cells, and so you can actually put them all at the same. One of the reviewers in a past paper we got actually asked us to do that, but currently, the way that the analysis, the algorithms are done, they already include that. So you can basically do a subset, 1,000 cells from this group, that group, and that group, and validate your approach. Another way to actually do that, it's not so much in terms of number of cells, but in terms of patient-driven specificity. So if, for example, one patient would actually lead to a differential expression pattern just from one patient, that is also removed, because we don't want something that is just because of one patient alone. So we do something that we call, leave one out. So we do the analysis several times by removing one patient and seeing if the differential expression is similar. Yes. Yeah. So they have to be both good. Yeah. So we own all the cells that are here have both ATAC and expression. So yeah, so we use to identify the cell types, we actually use gene markers that are typically annotated for. Oh, actually, can I just do this? Let me go. That are known to be important in different cell types. You can see here, for example, these T cells all are high in these different cell types. And so when you actually do this analysis, you can get a proportion map or plot like this, where you can actually see for each patient how many cells are the epithelia, which in this case are non-malignant on the top for the fallopian tube and malignant in the bottom for the cancer cells. And you can see right away that, for most part, the tumor itself is very rich in epithelial cells, whereas the fallopian tube actually has a lot of different cell types. And that, that's something that people have been interested in understanding, because precisely of the immunosuppressive environment that ovarian cancer is known to have. So this is the first, once we look at all the different cell types, we wanted to narrow down to epithelial cells, right? So we wanted to know what happens in the cells that actually get transformed into cancer. And so we put all the epithelial cells together. In this case here, we actually use a method called Harmony to remove patient variability. So now we actually have all the epithelial cells clustered together from different patients. And then we wanted to actually find a specific pattern that potentially could tell us which ones are the green cells that I was illustrating before. And that was actually my initial hypothesis, that if we incorporate RNA expression and open chromatin, you will be able to really define much better what's happening in these cells and potentially define the drug-resistant cells. But that was not the case. You see that there's, they all cluster very nicely together, and then you can kind of like define specific subclusters if you go on the edges. And so the ciliated compartment is, as I said, one of the cell types of ovarian cancer. Secretory is the other one, and these are the ones that are thought to be more malignant. And you can actually see that, there's a lot of ciliated cells in the fallopian tube, but they kind of get lost in the tumor. And then also one interesting point here is the cycling with the cells that we so-called cycling cells, because they have a lot of cell cycling genes. And those are pretty much only present in the naive context, because of course, neoadjuvant chemotherapy really targets cells that are cycling. One important thing that we can do from this initial analysis is not just using expression, but then also using open chromatin to start inferring links of activity. So as I was telling you about how we are interested in how regulatory elements can affect expression, we can use ATAC-seq data to look at events of open chromatin associated with expression of a gene, and all those events piled together give us, for example, in ciliated cells, give us a map of potential enhancer regulation links that are represented here. This is, for example, for FOXJ1, which is a ciliated, highly expressed gene in ciliated cells, and this is for mesothelin. And you can start seeing like how the cells actually use the surroundings how the genes use the surrounding regions to activate specific genes. Yeah.
Is there always a correlation with open chromatin and gene expression? Have it in some cases. In most cases, it could be very interesting. Signs of
right.
And then my second part of the question is, what about?
Yeah, that's, well, now I'll start with the second one. The multiple enhancers is really the hardest part to deal with, especially, you know, when we're trying to infer activity. Now, there is a part of the story where we're going to try to do basically calculate if we have 5 enhancers that are activating, 2 that are repressing, is that really, you know, correlated with the outcome of the gene, right? Because we don't know. And, you know, most importantly, these are really sparse, this is a really sparse data set. So ATAC-seq from single cell, we get for each cell 3,000 to 5,000 open chromatin sites. So it's very, very sparse. The only way we actually get to do some of the analysis is by pulling the cells together. So pseudo bulk ATAC-seq. And to answer your first question, I mean, the reality is it's not always true. Like, so open chromatin and expression are not always correlated. And that's something that we also have to deal with in terms of analysis, right? Because yeah, it gets really fascinating and complicated. So the first thing that we did, I'm going to kind of like go a little faster on this analysis, this initial analysis. We were trying to basically understand what are the differences between fallopian tubes, so non-malignant and malignant. We found a few genes that people had already described as important in the development of ovarian cancer or high-grade serous, namely MECON, WT1, PAX8. They are, you know, drivers of ovarian cancer. And then, not surprisingly, if you look at, you know, pathway analysis of this differential expression profile, you see that, you know, a lot of things are associated with checkpoint, cell cycling, mitotic, E2F targets, and MYC targets. So all of this makes sense because these cells are proliferating. And then what we actually wanted to learn from this this multi-omic set is like not just the genes that are expressed, but maybe potentially how they get regulated. And so what we do here, we use this ATAC-seq clustering. So we're looking at open chromatin regions. Again, this is the pseudobulk signal. And then we look at DNA binding events. These are all bioinformatics. So this is not something that actually is collected from these cells. But we know that, for example, DNA binding factor 1 binds here and here, and there's 2 open chromatin sites on that region. So this guy gets, you know, out of 3 gets a score of 2. And we can do this for all the different transcription factors or DNA binding factors from our, you know, curated data sets. And so you basically start giving to each cell a score for each transcription factor, and then you try to correlate them in terms of like, are these transcription factors almost always correlated with each other? And that's what this big plot is showing here. Everything that it's in green basically means that transcription factors tend to, every line here is a transcription factor. And so if it's really red, means that these transcription factors more likely are acting together in some shape or form to, you know, activate genes or repress genes. And, you know, not to make this very complicated, we did see some interesting features. So, for example, the MECON, PAX8, and WT1 typically are together with SOX17, which is also an important factor in ovarian cancer. And then once we get to the naive compartment, they actually get separated. So this really highlights the fact that, you know, potentially there's a lot of things in transcription factors that are changing, and that's what we try to understand in order to tackle drug resistance. And so, you know, this is another way to see that correlation. Here, we're basically mapping all the DNA binding factors and their potential, the genes that are regulating. And so this is kind of like systems biology approach, but in this case, coming exactly from ATAC-seq events and RNA expression. And so on this side, we have, for example, transcription factors that are known or that from our data seem to be important specifically in fallopian tube. On this side are the ones that are specific to high-grade serous, and then the ones in the middle are the nodes that seem to be common. And so even though there's a lot of things that are common, what is actually very interesting is that some of these transcription factors actually change from activating to repressing. And that's what basically I'm trying to highlight here in the bottom. This is green is for transcription factor gene activation, and red is for transcription factor gene repression. And you can see that in tumor, there's a lot more events in repression, which is also highlighted here. So here I'm looking at all the transcription factors with the scores for regulation of genes, and you can see that they are all lined up. So for this batch 2, initially is mostly an activating gene, but then in tumor it becomes a repressor. So we want to look at these variabilities to try to understand the oncogenic process. Yes.
Normal cells? Yeah, on that cluster.
This guy.
Yeah.
Mm-hmm.
But then in the cancer sample.
Yeah, exactly. Yeah, so those are the
Like that, because those are regions that are all.
So what you're seeing here is open chromatin regions that are highly bound by RFX2, and then if they're close by a gene that is active, they are called activators, and if they're close to a gene that is repressive, they are called repressive. And this is only possible because we have 10 different cell types, and so you can actually see for that same open chromatin region, does it always connect with the gene being on or off, right? So that's kind of like the approach that we take for this. This is, we didn't develop this as a figure algorithm that we've been using. Well, not yet. We don't explain that. So for example, if one is interested in that specific gene, what you want to do is, for example, try to understand what genes are regulated by RFX2 in fallopian tube, and then which genes are regulated by in the tumor, and then look at expression, do gain of function, loss of function, and see how that works. So that's at the specific level.
Cells, so it includes every cell that you were able to.
So what I'm showing here, this is in the fallopian tube, the epithelial cells, and the high-grade serous. But the way we annotate whether they're activating or repressive is by using the fact that, for example, in the T cell, the open chromatin region and the gene are not associated with activation, but they are in high-grade serous. So it's really kind of like a very heavy bioinformatic approach to really put these things together. So then we wanted to understand, again, coming back to the idea that can we actually know what the drug-resistant cells are like? We basically decided to take advantage of the fact that we had 3 samples that actually had been treated with chemotherapy, and this we called the residual disease profile. And so we have three patients with residual disease and 6 that are, normal high-grade serous that basically would be treated after debulking surgery. First, we look at RNA expression, and I'm just gonna browse through this a little quickly so that we get to the punchline. So looking at expression, differential expression analysis between naive and residual or NACT, we see obviously a lot of different genes. We actually try to map them to known prognostic genes that people have been publishing in the literature, and you can see that we actually color-coded them so that you can kind of have a sense of like how a lot of these genes actually are in different places. The ones that are prognostic should be on the right side and they're on the left. And so for the expression itself, we couldn't really find something that was very specific. But then when we actually look at open chromatin regions, we actually saw a very strong shift of enrichment in the residual disease. And this was even more exciting because a lot of the transcription factors that we knew are important in high-grade serous were also here: PAX8, SOX17, MECOM, estrogen, and et cetera. And also we saw some stem cell markers. SOX2, I don't show it here, but it's one of those. And so we were kind of intrigued by this idea that the epigenome of these residual disease cells had already some kind of pattern that we could look into. But then what we wanted to really do is to see if this residual disease signature was also present in, or was also predictive of resistance or sensitivity. And we call this the persistive signature because we're looking at the cells that persisted. So when we now go back to our naive tumors, we actually have two different groups. Three patients that actually responded very well, so we call them sensitive, and three patients that where the disease came back within 6 months, and so these are called the resistant tumors. And so we could actually now have a good cohort of 3 patients on each side that are either sensitive or resistant. Is the expression or the chromatin patterns going to help explain whether they're going to be sensitive or resistant? So that's what we did here. We compare what's up in NACT versus what's up in the resistant. You see the overlap here. We also did this for sensitive, for the downregulation. We didn't really see much there. But then what was really striking is that when you compare the residual disease and then the resistant, the overlap is very striking. So a lot of the regions or the open chromatin patterns that we saw in the residual disease were present in already patients that are resistant. So we try to shorten this list a little bit. And so we basically looked at things that are, the score is very high in the residual disease versus the ones that are low in the naive. And then we made sure that the transcription factors or DNA binding factors that we are looking at in this signature are expressed in at least 1% of the cells. And this came down to 178 DNA binding factors. What we can do after that is we do exactly the same clustering approach that we did. So we give a score for each transcription factor, in this case 178, and you can basically then make an average score and give what we call the persistent signature score per cell. This is just a overview of all the transcription factors and chromatin regulators that were part of this list, the 178. And then when we actually go back to our epithelial clusters and you try to map or you map the signature score, you see that the sensitive has the lowest, the NACT obviously has the highest, but also the resistant is actually in between, and it's significantly up from the sensitive. So this really means that somehow the score in the resistant cells is actually higher. We were interested in trying to understand whether this score really meant something in terms of expression. So we looked at a longitudinal study in RNA-seq that actually shows that in patients that the tumors resisted, there's this stress-associated signature that includes a lot of the genes that we actually also find regulated by our signature. And they can actually show that this stress-associated signature, comprising of 35 genes, actually is predictive of prognosis, has a prognostic power. So what we did then was to try to do, again, related to the question before, try to understand whether these networks of enhancers and genes really help explain a little bit of these, for example, stress-associated genes. So we pulled down all the gene lists from this paper, and it also included quiescence-associated, and try to see, can our signature explain some of these signatures? And this is the proportion of the genes that are from the signature that is explained by our signature of transcription factors. So this is kind of just representing what I'm trying to show here in green. Green means that there's a gene that basically has a lot of links, activating links towards this gene locus. Red means that most of the links are repressive. Yellow means that it's kind of even. So you have 3 enhancers that are repressive, 3 enhancers that are activating. And then the gray is basically genes that we do not find any links that are in our list. Doesn't mean that they're not regulated, but it's just not explained by our list. And we were pleased to see that stress-associated had a lot of associations with our signature, meaning that this epigenetic propensity for stress response is already present before chemotherapy, and we can show in specific genes how that actually looks like. So genes that are upregulated in the resistant are all associated with some of the genes that some of the transcription factors that we identified. And this, at least for me, is kind of like the most striking comparison, is because at this point we were really puzzled in terms of why do we have so many transcription factors or DNA binding? So 178 is a very big signature. Is this really do they always have to work together? Are we pulling 178 because maybe we have only 3 patients on each arm? That's possible too. But we wanted to at least understand if these 178 were potentially working together. And so if you compare the naive So before chemotherapy and patients that actually had seen chemotherapy, you see that the signature becomes really strikingly correlated. So all the transcription factors or DNA binding factors from the signature are here, this barcode on the top. And you can see that here, it's very mostly dispersed. But then in our after residual disease, you actually see that they kind of clustered together, which potentially means that they actually are working together towards survival of these cells. And because, as I mentioned, we didn't have a longitudinal study or samples, we decided to take this in vivo, and we used patient-derived xenografts that where basically an ovarian cancer tumor is taken out of the bulking surgery and put into a mouse and grown as a tumor in these immunosuppressed mice. And so what we actually did was choose 3 different tumor patient samples. One that is sensitive, so when you treat it with chemotherapy in the PDX, the tumor shrinks. One that we call stable, which is a situation where basically the tumor doesn't grow but also doesn't disappear. And then one other model where we call it resistant, because regardless of the treatment, it keeps on growing just like normal. We did something here to try to understand whether we were actually picking up specific clones, and that's regarding your question. We actually can use mitochondrial DNA because it's the mitochondrial genome is so small. We can use mitochondrial DNA and we have enough coverage to look at specific variants. And by looking at specific variants for each tumor, you can try to see whether there's a subclone that actually was arising from the tumor. And overall, we think that the majority of the tumors look pretty much alike. And so what we did next was looking at our epithelial cells, score them, and basically here showing exactly a very similar pattern to the human samples. The sensitive has the lowest score, the stable is in the middle, and then the resistant has the highest. But then very interestingly, when we actually look at, this is, sorry, I forgot to mention, this is the score before the tumors got treated with chemotherapy. When you actually look at before and after, the tumor that is sensitive actually grew, the score actually went up, and that's also significant. And you can see a very similar pattern that we saw in the patient samples. There's a widespread of correlation in untreated, but then in the residual disease, these 178 genes tend to cluster together. Whereas in the tumor that is actually resistant, this didn't really change at all. So it was potentially inherently already resistant, and that's at least how we interpret it, because you can see the score was already high and didn't really change much. So that's kind of like our story for today. We basically have been trying to use this multi-omic approach to try to understand what we call these transcriptional dependencies. We believe that we identified a so-called persister signature that is epigenetic, because by just looking at expression, we can't really see or we can't really predict whether the tumor is going to be responding to chemotherapy or not. And this signature is associated with stress response and stem cell regulators. So one could also potentially call it a cancer stem cell signature. And then also kind of interesting, and this is what we're really going to follow up next, is to understand this rewiring of transcription factors. And so you can pick your favorite transcription factors and try to really figure out how they are actually working in the normal context and then change into this. And so, the goal eventually is, to do what computers always now annoyingly do all the time to us, is like to prove that we are human. We have to click on these things just to, they understand that you actually are behind the screen. But the goal for us is also to do the same thing. If I were given a tumor and I would be analyzing every single cell, could I actually tell which cells are going to be resisting or not? We're not there yet, but if we pull them together, we do see these signatures that potentially could be interesting and targetable. So just to thank you guys, this is my lab. I highlighted the work from Wazim, which is our computational biologist, Mihai, who is our PhD student, and Leticia was initially in the work, but she already left our lab. And so also for the funding and of course for the patient samples and their families. Thank you for your attention.
Thank you for the talk. That was very impressive work. I have 2 questions. I think your first one is, it seems to me, based on what you presented, that there is a lot of transcriptional heterogeneity between the patients. That's your biggest contribution in your UMAP analysis.
But then when you presented the epigenetic signature, it seems to be less variant. Is that correct? That the tumors between the epigenetic signature between the patients is more homogeneous than the transcriptional signature of the patients?
Currently, yes, but you have to think about the fact that, this is, so the analysis of the gene, the expression is kind of relatively easy, because with RNA expression, even though single cell doesn't have enough depth, you can count, 100 molecules for gene X, 50 molecules for gene Y. In the analysis that I'm looking at by ATAC-seq, what we're actually looking at is events, and they're very sparse. There's like 3,000 per cell. Events that are closely associated with binding of a transcription factor. So it is possible that the reason why we see it so homogeneous is because the data is a sum of all these together, and we don't really get to see the heterogeneity that each patient by expression actually shows. I don't know if I make myself understand, but it's really, I think it actually comes more from the data. What I'm really surprised is, like, do we really need those 178, right? Like, that's really the question that still is in the back of my mind. Like, I don't think, I actually think if we are able to expand this cohort a little bit more, we'll probably be able to refine, and actually we'll probably be able to actually find signatures that are important in homologous recombinant deficient and proficient, because that's something that we still don't have.
Right. And my other question, I think you kind of answered at the end, but so you said that you haven't identified a population of cancer cells,
the tumor,
the naive patients, but in the two groups that were like the responders and non-responders, can you infer the proportion, if there's a difference in the proportion of the putative cancer stem cells based on the ones that are not responsive to the? Yeah,
so we started looking into, like, by patient, looking at the score and see, like, can this score really determine whether the cells are going to survive? The only way to do that currently is to use the PDX model, right? Because we have before and after. With patient samples, all we can say is that we find a lot more cancer stem cells in the patients that are going to be resistant than the patients that are not going to be, that are going to be sensitive. And that's really not very good, because as I was trying to, represent in that diagram, if you give me 1,000 cells, I still will not tell you which ones are going to be surviving, right? And that's really the challenge. Like, we see a signature if you put them together, and okay, this is higher here and low there, but I'm not able to, like, put them on the floor and say, oh yeah, this guy is. And that was kind of like my hypothesis. I was hoping that using a multi-omic approach that includes RNA and ATAC-seq, I would be able to see that, but I don't think we're there yet.
Thank you.
Thank you very much. Specific question on multi-omic. Have some kind of hierarchy of those transcription factors.
Right.
And my general question is, for me, it's clear why you started with transcription. But we know there are multiple overlays. About what are your thoughts concerning that? Example, next step will be the partners. One of our recent interests, repetitive sequences.
We had the same exactly. Yeah, so I can start with the second question. So definitely in my lab, we are currently performing the genome sequencing to actually know whether the tumors that we have are homologous recombinant deficient or proficient, because that will change obviously the analysis. And then the other one is we're performing DNA methylation to actually see, obviously not in single cell, because currently it's really not that feasible, but we're trying to see if some of these patterns have something to do with DNA methylation, because so one of my students is actually looking at disposable elements precisely because he's interested in trying to see what other modalities. So clearly there's more modalities to look at than the genome, and the metallome is definitely something that we need to look into. For the first one, yeah, I mean, I really like the idea of prioritizing. I mean, I basically told my postdoc, I mean, he was so tired of hearing me say, like, you know, I want this list to be shortened by at least 50. And we try a lot of different things. We try different classes, if there's something like very specific. I think at this point is really a question of number of patients. I believe if we have more, we probably would be able to kind of make the list a little shorter. Because the data, like a lot of the data that we have, for example, we have a lot of June C, June D, June B. It's possible that only one does the work, right? But we don't know which one, so we can't remove it from the signature. Because we did that. We did some that are kind of like isoforms that could probably doing together. We try to remove all of them. We didn't do, you know, specifically for, you know, this class or the other. We just did it globally, and we couldn't really find, you know, prioritize. But, you know, there's definitely room for that in the further analysis.
So regarding the question that I asked you at the beginning, the data that you presented suggests that apparently you have a population that is already able to resist the chemotherapy, right? Mm-hmm. You have the data to address that particular question from a different perspective than the one that you use. There is a statistical test that is called the fluctuation test that was the Nobel Prize for Lurie and Delbruck back many years ago. And basically what you will have to check is if those cells that you call persisters, or I think that's the name, if the frequency that you see in each one of your patients, because you have about, I think, 5,000 or 2,000 for each one of your patients, cells, if the frequency of those persistence phenotype or transcriptions is the same in all your patients,
right,
that will be consistent with the idea that this is
predisposition,
a selective phenotype, that the cells are already there and the chemotherapy is selecting for them. But if you don't see the same frequency of these cells in each one of the patients, so the conclusion that you are
is an adaptive
right now one.
Well, I mean, because the thing is, like, I actually open to both, because as you saw from the PDX, the sensitive tumor adapted and gained score. The persistent score was higher. So it is that even though the tumor was, you know, regressing, the cells were reacting to it, showing the same signature that we actually had seen. So I actually don't think it's one or the other. I think potentially in some patients it's going to be very, you know, obvious that they have this signature already present. In some of them, they actually might be kind of in between. And depending on how many cells have that signature, they will be able to either resist or die. But then, you know, and also, I mean, I think that's a great suggestion. We'll try to follow up on that. But this is also, you know, very specific to chemotherapy. Maybe this epigenetic signature is different for PARP inhibitors, right? And so then we're like, so are the cells, because the cell cannot be ready for everything, right? You know, the tumor cell obviously has gained selective advantage over the normal condition, but obviously there's going to be, you know, tumor cells that will be responding very well to PARP inhibitors and others don't. So the signature might actually be different depending on the treatment that you give, and so it makes it even more complicated, right? In
that scenario, you don't have an immune system.
Yeah, right. Yeah. I mean, that's a great point. I mean, and that's one of the reasons why, you know, having longitudinal data would be ideal. And then the other one is to look at the signature first in the patient samples and not the PDXs, because we had the PDXs before, but I was really keen on trying to understand whether this persistent signature has something to do with the microenvironment, and I think it does for sure. It's nice that we see the signature there. Actually, in the beginning, I didn't even expect to see it, but I'm pretty sure it will have a lot of you know, complicating factors once you have a mouse model that, you know, is syngeneic, and we actually have one now that we will potentially try. But I do think, because I mean, the tumor doesn't grow on its own, right? Like, it has so many things and so many complicating factors, and that's why studying the patient samples, at least for me, it's a good start for discovery.
germ cells or any other cell type in the ovaries?
Yeah, so there are specific ovarian tumors that are from the germ cells, and those are rare. We didn't really put in this cohort, but they do exist. There's also clear cell. Yeah, they're very rare. So the germ cells actually are pretty lethal, but they're relatively rare. That it's kind of hard to actually study them and even have a cohort for those. But yeah, they do exist.
In the cancer cell, I remember that you have some genes like KLS, not SOX2 or 4.
SOX2 I actually have it, but we didn't highlight it because the SOX2 is actually in this signature. So SOX2 in the big slide, it comes up.
Or any other related.
OCT4 does not make it because we actually don't detect it. So by RNA-seq, by the single cell portion, we get it out.
That's a lot in the field of cancer cells. They try to argue that there is transformation, turns on a pluripotency-like program that drives proliferation and cancer.
Yeah.
What's your view on that?
So actually, I kind of went a little fast on when we were looking at what genes this signature might be regulating. I actually think that even though we see stem cell markers, and people tend to think, especially, I came from the reprogramming field, that, stem cell markers are very highly proliferative. I think, for example, SOX2, and it has been shown in other systems, not ovarian cancer, is actually, in this case, a stem cell marker, but it induces senescence. So it actually keeps the cells growing less. And that also has a lot to do with the stress response, right? Like, we know that these cells have to stay somewhere for a period of time until they actually come back, right? So we can't really think of it as like when it's a very, malignant disease, a lot of the cells actually might not be, growing and proliferating as we think they are. They actually probably enter a state that allows them to stay away from the immune system, or, just stay low for a while. And, because treatment for chemotherapy, for example, can last, 3 months. So I think it actually is there's a little bit of both. And so these stem cell markers actually can have dual effects. And that also comes back to the idea that in different contexts, a transcription factor acts activating some genes, and another it can actually repress them. Because in embryonic stem cells, for sure, SOX2 acts as, very importantly in the proliferation of cells.
And it's very interesting because in that sense, could have something to do with differentiation or reprogramming.
Yep.
And then maybe to ask you, what about the heterochromatinization of terms of differentiation and reprogramming?
Right. Yeah. So I mean, we haven't looked globally, because I guess we could start looking at H3K9 trimethylation marks and see how in these patients. I mean, we started doing cut and run for some of these, so that could be potentially something that we can look at. But we also actually are trying to do high C approaches to see how, I mean, in my mind, like this potential quiescent state or driven by these 178, are they actually, using the same topological domains? I mean, is this something that actually is involved in the, in the 3D? And I do think it is. So I wouldn't be surprised if we find something.
Can I ask a final one? Just a general question of your signature. You see like between repressors and activators, is there like a
trend?
A trend?
No, because they change, right? So I mean, when I showed in the beginning that the plot with the green and the red, and they actually change a lot. So there's a little bit. I think there's more repressive activity in the concept of cancer, and that could be related to stress response, quiescence, like these kind of mechanisms. But I mean, the trend is it's hard because they actually change a lot, and we would need. I mean, at this point, we need to validate them one by one, I guess, or as like a pulled screen to try to see if their effect is mostly activating or repressing.
And just one other one. You must have a lot of accessibility peaks that maybe they don't have a correlation with up and down. Because those might represent some post phase that might be very important
right.
for the disease or the resistance. Have you have a look?
No, I mean, I think this is a great suggestion. I think we should definitely look. I mean, initially you try to explain as much as you can with things that are correlated, but I definitely think that looking at the things that are not explained by open chromatin, or they're not associated with the things that we are expecting, would actually be very interesting, for sure. Thank you for the suggestion. All right.
Now that I'm listening to all these questions, it came to me, this idea, it's a more basic question. I mean, when you study the structure of the chromatin, it makes sense to measure transcription and all those things. But in terms of the function of those transcripts, as well as the change in the chromatin for the cellular phenotype, there is this now more than 10 or 15 years old experiments that show that the transcription and translation in eukaryotes are not related.
Right.
So, what's your perspective on this? Because you seem to make some assumptions about the phenotype consequences of this.
Right.
You know?
Yes, I mean, obviously this is knowing whether this signature would affect actually the protein at the protein level. For example, the genes that are associated with stress would be obviously very important. Currently, it's really hard because single cell approaches to look at protein are really complicated. And so if I were to come to you with my signature at the single cell level, it would be really hard for us now to actually see whether those markers would actually be expressed at a protein level. But there's cell surface markers, and so we can start looking at, for example, genes that we know are associated, or people have shown are associated with cancer stem cells. We haven't, by expression, we actually don't see that. So it comes back to your point. I mean, we don't really know, and this is 10% of the picture at this point, probably.
Thank you very much.
Thank you.